Pentoses and hexoses, or monosaccharides with 5 and 6 carbons, primarily are in the ring form when they are dissolved in solution. So that means when they're in the bloodstream or when they're in cells. So the majority of biological situations where we're looking at monosaccharides, they're going to exist in the ring form. The hydroxyl group that is on the anomeric carbon in the ring form is very, very reactive. And this hydroxyl group on the anomeric carbon can react with another hydroxyl group on another molecule. And when those two react, a glycosidic bond forms as well as a water molecule. So anytime two monosaccharides are joined together to form a disaccharide or higher polysaccharide, they are joined together by a glycosidic bond. So let's look at the formation of a glycosidic bond between two monosaccharides, and on the slide we have the Haworth projection of 2-alpha-D-glucose, and we see the hydroxyl group on the anomeric carbon is below the plane, so they're both alpha-D-glucoses. So the anomeric carbon on the left, the molecule on the left, has a hydroxyl group that is in red. And I know this is the anomeric carbon because it is the only carbon that is bonded to two oxygens. So this hydroxyl group is very reactive, and it will react with the hydrogen of another hydroxyl group to form a water molecule, and then this oxygen on the hydroxyl group that the anomeric carbon hydroxyl reacted with, this oxygen will form a bond with the anomeric carbon, and that's what's known as the glycosidic bond. So when two alpha-D-glucoses form a glycosidic bond, the glycosidic bond, which is highlighted in blue here, is also in the alpha position. So the glycosidic bond is between the anomeric carbon and the oxygen from the alcohol functionality on whatever other molecule that anomeric hydroxyl group reacted with. So our glycosidic bond is in the alpha position. It's below the plane, opposite the side of C6 of the molecule that the anomeric carbon is on.